and are a potential replacement for some petrochemically plastics. And I think the idea of this, the importance of where we get materials from, provenance, has probably never been so important. And if we produce new biopolymers or bioplastics that do not use petrochemical oils, then we're getting somewhere. So I hope that this sparks your interest, some of the students, and you know, be in touch with Pooja. She's very approachable, and see what she's doing. She very kindly came and showed me, brought a whole bunch of samples once to the office, which is terrific, like these bits of stuff growing in jars. Really amazing. So thanks so much for joining us. I know that Pooja's a bit worried that a presentation might be a bit, I think you called it a bit life sciencey, so it's going to be some science tonight, but you're very, I saw you talk before, you were incredibly clear about describing what you were doing, so so I think we can cope as architecture students. I hope we can. Anyways, thanks very much.
Thanks, William. Thank you for your kind introductions. I'm just going to share my screen and start the presentation. Okay. Please do let me know if you're able to see my slides, William. I won't be able to see the chat, so you'll have to tell me.
Yes, we can see your slides. That's perfect. Okay.
So should I start?
Yes, you should start. And I think if there's any questions, then maybe put some stuff in the chat, and then we can answer them at the end, if that's okay. Great, thank you.
Okay. So good evening, everybody. Thank you for joining this session. I'm Pooja Basnett, and I'm a senior lecturer in the School of Life Sciences. So I am from Sustainable Biotechnology Research Group. This evening, I'm going to share a little bit about my research, which is focused on bacterial polymers. So we're all aware of plastic pollution and its fundamental links with climate change. If we consider the life cycle of a plastic product, how it is made, so it's derived from fossil reserves, how it's manufactured, how is it used in different sectors, majority of it being used in packaging sector, and its end-of-life options. So where does it end? Incineration, recycling, landfill environment. So if we consider each of these different steps, a significant amount of carbon dioxide, which is one of the main greenhouse gases, is released, which accounts for the global annual carbon budget. Now, which is worrying, right? There's another interesting article that was published in 2022 by a group of researchers in Italy. They reported the presence of microplastics in human placenta. So they collected 6 placentas from pregnant women, of course, with their consent. And what did they find? They found these microplastics. So microplastics are smaller fragments of plastic waste. And in 4 of those placenta, they identified these microplastics. There were 12 pieces of these microplastics that were found, out of which they could identify 3 of them, and they were found to be polypropylene. So polypropylene is the kind of plastic that we use in our bags to make yogurt containers, for example. They couldn't identify the other 9 fragments, but what they could identify was the pigment, which came from man-made coatings, paints, polymers, cosmetics, and personal care products. Now, what does this indicate? What does this suggest? That these plastic waste, microplastics, they transition, they move from one ecosystem to the other, which is very worrying from human health point of view, from the environment point of view. So there are kind of concerted efforts being made globally to tackle this plastic issue, be it policy initiatives or improving recycling facilities or by reducing the use of single-use plastics. We are one of the many research groups that focuses on building sustainable, environmentally friendly alternatives to replace synthetic plastics. Now, how do we do that? We do that by developing these polymers, these plastics that have properties that are very similar to synthetic plastics. In fact, their properties are identical to polypropylene, but they are produced by bacteria. They're produced by bacteria, and they are produced using renewable carbon feedstock. Now, what are these? Okay. Now, what are these bacterial polymers called? They are called polyhydroxyalkanoates. I know it's a long name, but we call them polyhydroxyalkanoates or PHAs. These are a family of biopolymers or polymers produced by bacteria. In nature, there are a range of bacterial species that are able to produce these plastics. They are produced intracellularly, which means that they are produced or accumulated within the bacterial cells. I'm not sure if you can see my arrow, but if you look at this image, the second image at the bottom, and you see these white bits, these are plastics accumulated inside bacterial cells. Now, how and why and how do these bacteria accumulate these plastics within them? We feed these bacterial cells with a lot of carbohydrates. So we provide a lot of carbohydrates to them, and then we limit one of the other nutrients. It could be nitrogen or phosphorus. So we kind of we artificially create a very stressed environment for the bacteria. And what does the bacteria do? It starts to accumulate these energy compounds, energy reserve that it can use later on. But what do? We kill this bacteria, we extract the plastic, and we use it for different applications. Now, these plastics, they can be broadly classified into two different types: short chain length and medium chain length. Now, well, these bacterial plastics are basically polymers, right? Which is formed by joining of monomer units. Now, depending on the number of carbon atoms, and if you look at this image here, you see C4, C5, C6, and C7, all the way to C12. What does that mean? It indicates the number of carbon atoms in each of those monomers that join together to form this polymer. Now, those monomers that have 4 to 5 carbon atoms, they are classified as short chain length PHA. Those with 6 to 12 carbon atoms are known to be medium chain length PHA. Now, why is this important? It's because the physical properties of this material depends on its type. So short chain length PHAs, they're very brittle, they are crystalline in nature, therefore they have a very high melting temperature, whereas medium chain length PHA, they're very elastomeric in nature, they have a lower melting temperature, they have lower crystallinity. Now, why am I talking to you about these bacterial polymers at 6 p.m. today? It's because of its attractive properties. They are biodegradable in nature, right? So they degrade in the environment. In the environment, there are several microorganisms that are able to release these enzymes called PHA depolymerases. So any bacteria that's able to release these enzymes, and there are many, they're able to degrade these plastics. So unlike synthetic plastics that we buy from supermarkets, we use for packaging, water bottles, etc., they cannot be degraded in the environment. There are very few bacterial strains that have been identified that can degrade synthetic plastics, but not fully. They are biocompatible, which means they are non-toxic to human. They degrade via surface erosion, which means if you are intending to use them as an implant within the human body, they degrade in a controlled manner. So how does the bacteria accumulate these PHAs? What is the biosynthetic pathway? Please don't be scared looking at this very complicated biochemical pathways. So depending on the type of microorganism we use, depending on the type of carbon depending on the carbon feedstock that we give the bacteria, all these different kinds of metabolic pathways occur inside the body of the microorganism, which leads to the accumulation of these plastics. Now, on the right-hand side, you can see different end-of-life options of these materials. So they can be recycled, incinerated, or burned. They can be composted, but most importantly, they can be biodegraded. So they can be degraded by microorganisms in the environment. Now, how do we produce these bacterial polymers or bacterial plastics or biopolymers, whatever you call it? We produce them in a lab environment. The process is called fermentation. It's very similar to how people brew beer. I'm not sure if any of you are familiar with the brewing process, but this is very similar to that. So we take some bacterial colonies. If you look at this first picture here, you see these round colonies. These are microorganisms growing on a plate. We take those, we prepare a broth, which is also called the starting culture or the seed culture. We culture these bacterial cells in a vessel, which is also called bioreactor. So bioreactor kind of provides a very controlled environment for these bacterial cells to grow. We feed the bacteria with lots of carbohydrates. We limit one of the nutrients, it could be nitrogen, it could be phosphorus, and we grow them for 48 hours. Right, so this is a picture of the bioreactor, which is housed in one of our fermentation suite at the Cavendish campus. If anyone's interested, I can give you a little tour of the lab. So what do? We culture these bacteria in these bioreactors. If you look at this graph here, it looks complicated, but it's basically monitoring the process of fermentation. So we look at different process parameters. We measure the concentration of nitrogen because it triggers the accumulation of PHAs within the bacterial cells. So we measure the amount of nitrogen throughout the process of fermentation. We also measure pH. We also measure optical density. Optical density basically measuring the turbidity of the liquid in the bioreactor. So what does that do? It indicates whether the bacterial cells are growing or it's dying. So as bacterial cells grow, the optical density increases and it starts to die, it kind of starts dropping. So we monitor this fermentation process. On the right hand side, if you see, this is a scanning electron micrograph of the microorganisms that we use to produce these plastics. After 48 hours, we centrifuge and we retrieve these bacterial cells. Like I mentioned earlier, these are intracellular compounds, which means that they accumulate inside the body of the microorganism. These brown powder-like things are basically bacterial cells. They are dried in vacuum condition, and they are crushed and ready to be used for the downstream processing. Now, the picture that you see here is a Soxhlet apparatus. It's a very old chemistry equipment. So we are still using that to extract polymer from inside this biomass. At the moment, we are exploring the use of environmentally friendly organic solvents, but we want to move away from the use of solvents and explore other ways to extract these polymers from bacterial cells. I have a PhD student currently in the lab who's focused on sustainable downstream processing. And this is the picture of a very elastomeric sticky polymer that we produced. We wanted to use that for developing coatings, for example. Now this is, this is just an overview of how we produce PHAs. Now once PHA is produced in the lab, what's the next step? So these PHAs or bacterial plastics or polymers are characterized. This is to confirm that what we've produced is PHA. So we do FTIR analysis. So we pass infrared light through our polymer sample, and then we look for signature peaks, characteristic peaks. We also do gas chromatography analysis. It's another technique to identify individual monomer units. So we're able to kind of confirm the presence of PHA and also identify the name of the polymer. We also send our samples off to chemistry department at UCL for NMR, nuclear magnetic spectroscopy analysis. This is to identify the structure of the polymer. Now, if the intention is to use these polymers for different applications, we need to process them using different techniques, and that could involve the use of heat. So what we do is we characterize their thermal properties using differential scanning calorimeter, which is in the lab. So we kind of identify their melting temperature, their glass transition temperature, their crystallization temperature, etc. We also look at their mechanical properties. So again, depending on the type of application we want to use these bacterial plastics or biopolymers for, we have a tensile testing machine in the lab. So we look at 3 different properties: tensile strength, Young's modulus, which is the measure of the stiffness of the material, elongation at break, which is the measure of elasticity. I'm sure you all are already aware of that. So if you look at this table here, you can see 4 different types of bacterial plastics. So polyhydroxyalkanoates, like I mentioned earlier, is a family of biopolymers. So it's not just one type of plastic, but a range of different types of plastics with various properties. So just as an example here, PHA1 has an elongation at break value of 1200, which is highly elastomeric in nature in our bacterial polymer world. And if you compare that with PHA4, which is again another type of PHA, the elongation at break value is 2.4, which is a very brittle material. And similarly, if you look at the thermal properties, you see melting temperature of 42 degrees, and then you can also have a polymer with melting temperature of 171 degrees. So now, depending on the type of application you want to focus on, you can choose to work with any of these different polymers. What you could also do is blend these polymers or form composites with another materials. Again, it totally depends on the type of application you would like to explore. Now, based on literature, PHAs have been used for tissue engineering application, which is basically a process of creating human tissue outside of the human body. For targeted drug delivery applications, where they've used these bacterial plastics as carriers for drugs and active compounds. They've used these bacterial plastics to develop medical devices. For example, one of the first PHA-based medical product to be commercialized was sutures. They received FDA approval in 2009, I think, and also CE marking. The other applications that they've been used for is in wastewater treatment, in food packaging, as biofuel additives, etc. Now, what are some of the challenges associated with the production and commercialization of these bacterial polymers? High production cost. So the type of carbon feedstock that we provide the bacteria with is expensive. Low productivity. Now, this is all in comparison with synthetic plastics, right, which are produced using fossil reserves. They are chemically synthesized in the lab, right? Lack of infrastructure. Now, again, in comparison to synthetic plastics, the infrastructure we have for bio-based products is inferior. So, what are we doing about it? We are focusing on economical and sustainable production of these bioplastics from bacteria. We are trying to find ways to bring the cost down so that we can use them not just for medical applications, but we can also use them for large-scale applications. For example, packaging, for example, in building materials. But in terms of price, we are nowhere near synthetic plastics. They sell a kg of PET for 50 cents, whereas we are selling 100 grams of PHAs for almost 100 pounds. And there's simply no comparison at the moment. Even when we compare the production capacities, 2 million tons of synthetic plastics are produced every year. And, if we compare that with PHAs, there's simply, there's just no comparison. Now, if we are really serious about replacing synthetic plastics using bio-based materials, what are the key areas that one needs to focus on? One is the cost, right? So how can we bring down this cost? So by using inexpensive waste materials. So we've used waste frying oil, for example, as feedstock to feed this bacteria to make these plastics. One of the issues is low productivity, so poor conversion of substrates to the final product. So we're looking at that as well. We are looking at metabolically engineering strains to improve the production capacity. We are trying to use novel, unique strains to increase the production of these bacterial plastics. So we're doing these different projects, basically. This slide is from one of my PhD students, and this has actually now been accepted for publication. We collaborated with this group in Italy, in Naples. They are this marine biodiversity group. they've isolated a very interesting, unique strain from the mouth of Sarno River in Gulf of Naples. And so Sarno basically is a polluted river, and this bacterial strain was isolated from the area which was heavily polluted with heavy metals, hydrocarbons, plastics, etc. So we've kind of taken this very novel extremophile, meaning microorganisms that are able to grow in extreme environments. We've taken that bacteria and used that to produce these plastics. We've tried different media compositions, and using this particular strain, we were able to produce at least 6 grams per liter of bacterial culture, which again in bacterial polymer world is very high. Almost 80% of the bacteria was able to accumulate these plastics. This is one of the media that gave the highest yield, actually. Okay, so now we've looked at what these bacterial polymers are, what their properties are, their types. We've also looked at how we produce them in the lab, and we've also looked at how we're trying to improve or scale up this process to make it competitive with synthetic plastics in terms of price. So now I'm going to talk to you a little bit about different medical applications that we've explored in the University of Westminster using these bacterial polymers. Okay, so one of the first projects that we were a part of was called NURIMP. It was a European Union funded project. It was a collaborative project with University of Westminster and other universities in the UK and also in Europe. The consortium was also made up of small and medium industries. The aim of this project was to use biopolymers and different manufacturing technologies to develop a peripheral nerve implant. So we're trying to focus on nerve regeneration using biopolymers and different manufacturing technologies. So just to give you a little bit of background on what peripheral nerve is and peripheral nerve injury is. So these peripheral nerves are links between brain and spinal cord and the rest of the body. Now, if this peripheral nerve is injured as a result of an accident or trauma, your brain loses ability to communicate with the rest of the body. Now, depending on the extent of the injury, it could pose mild discomfort to patients, or it could also lead to a lifelong impairment. So what did we do? Using biopolymers, we created a nerve conduit. And if you look at this image here on the screen carefully, you see this bridge that kind of joins a severed peripheral nerve together, right? So we used our bacterial polymer to create this bridge, or what we call nerve conduit, to allow guided regeneration of peripheral nerves. Now, how did we do that? So we took our bacterial polymer, we used extrusion system. Of course, this was in collaboration with other partners within the project, within the consortium. So we used different manufacturing technologies to create this really porous tubular structure using these bacterial plastics. We also have a dip molding machine in our lab, and we also used a dip molding machine to create these really nice tubular structures. What did we do next? We wanted to test. We of course did all the mechanical prototype testing. In addition to that, we wanted to establish that these bacterial polymer-based tubes were not toxic to humans. So we grew some stem cells on them. And if you look at these images here, they look green. Now, green indicates that they are alive. If there were dead cells on top of these tubes, it would be indicated with the red color. Now, we cultured these stem cells on top of these tubes to ensure that the cells were not dying. We wanted to ensure that the material was not releasing any kind of toxic byproducts that were killing these cells. So once we established that they were compatible with stem cells, with our collaborators, we implanted that in mice. So we implanted these tubes in mice for 12 weeks, and then on 12th week, We extracted the tube, and what did we see? We saw that the peripheral nerve had nicely regenerated. Now this consortium has a patent on this technology, on the use of this particular material and the manufacturing technology. So if any other company would be interested in making these nerve conduits, would have to license that patent from us. So this work has also been published, and I've put the link, at least the name of the link, as well as the name of the article on slides, if anyone of you would be interested to read. So that was one application of bacterial polymers. So we then moved on to another project where we used these polymers to create these nice fibrous structures. So for the first project, we created nice tubular structures and used that as a nerve implant. For this project, we created these nice fibrous structures. The technique that we used was called pressurized gyration process. It's very similar to how we make candy floss. So there's this cylindrical vessel with lots of pores around it. So we apply pressure and agitation, and then we collect these fibers. I have put the optical image of the fiber here, the first image, and the SEM or the scanning electron micrograph. You can see this really nice porous fibers. Now, what did we do with these fibers? We used them as a scaffold, as a three-dimensional scaffold to culture cells. Now, instead of growing human cells on a flat two-dimensional plastic surface, when we culture them on a three-dimensional scaffold, they're able to interact with each other. This environment is able to mimic the in vivo condition, what do we call it? Human body-like condition, basically. So we use them for different tissue engineering. So the idea was to create human tissue in the lab. Okay, so we focused on creating nerve tissue using these bacterial polymers as scaffold, providing a three-dimensional environment for the cells to interact with each other, for the nutrient supply to be distributed uniformly. So these colorful pictures basically are nerve cells very happily growing on these on this bacterial polymer scaffold. If you look at this image C is basically cells grown on two-dimensional flat plastic surface, whereas A and B are cells that were seeded on the bacterial polymer fibers. And you can see from the pictures here that they kind of followed the fibers and grew in a very uniform guided manner, which is very important for the function of these cells and tissues. The other experiment that we did was to look at bone tissue engineering, to create human bone outside of the body. So the first part of the experiment, you can see this little egg, and you see this little square on top of that egg. More than the experiment itself, to cut that little square on top of that egg was probably the most difficult part without breaking the egg. So what we were trying to do here was to create human tissue outside of the human body. It's very important to ensure that the material you're using as a scaffold is non-toxic first. Second, that it promotes blood vessel formation. So we placed this little polymer scaffold, or the bacterial polymer scaffold, on top of the membrane of a developing chick embryo and incubated that for 8 days, and we looked for blood vessel formation. And as you can see in this graph here, we were able to kind of report the presence of blood vessels. We then put this in mice again, and I'm not sure if you can see this image clearly, but you can see this little white arrows that shows the presence of collagen formation. Collagen is one of the major constituent of bone, so we could see the presence of collagen formation. We then also cultured heart cells on these fibers. Now, when a person suffers from myocardial infarction, part of your heart tissue dies. Now, the intention was to use this bacterial polymer as a cardiac patch to kind of heal those dead cells, basically. So what you see in this red color is the cardiac muscle cells. These are striated muscle cells that are responsible for heartbeat, for contractile forces in the intact heart. So what we saw was that these cells happily grew on these fibers. They in fact expressed phenotype which we find in normal healthy human body. And if I can, I'm not sure if this video is going to play, but so what we observed were these cells growing on these fibers, and you can see them beating. And this is in a petri dish outside of the human body. So which indicates that these bacterial polymers were conducive to grow these different human cells. This work has again been published in ACS Applied Material Science paper, and if you're interested, I can send you the link for that as well. Now we've got a very, very basic 3D printer in the lab. It does not compare to the fabrication lab that you have in your school. It's a very basic piece of equipment that we use to create three-dimensional structures. Again, we've 3D printed our bacterial polymer into different shapes. The idea here was to grow skin cells to create artificial skin outside of the human body. This was specially targeted for burn victims, etc. We were trying to see what would happen if we would be able to kind of grow patient's cell, outside of the human body, etc. So if you look at this, the image on the right-hand side, it's a layer of skin that's growing on this 3D printed material. And this is a video of how we kind of 3D printed our scaffold in the lab. Okay, so so far I've been talking about how to create these tissues outside of the human body. In this particular experiment, I'm not sure if you've ever heard of disease modeling, but instead of creating a healthy tissue outside of the human body, and actually thinking about it, why do we need to create human tissue outside of the human body, right? So one of the main aim is to restore the function of damaged tissue, and we're all aware of the issues with organ transplantation, donor shortages, issues with rejection, etc. So that's the intention with creating tissues outside in vitro. This is disease modeling basically. So here, instead of culturing a tissue outside of human body, here we're trying to create tumor outside of the human body. So instead of growing healthy cells, we were trying to grow kind of cancerous cells using these bacterial polymers again as a scaffold. So in this case, so far we've looked at 3D printed structures, we've looked at fibrous structures, we've looked at tubular structure. In this case, we created these 3D structures by simply kind of making a polymer solution, pouring them into a Teflon mold. It was almost like baking, the process of baking. So you take the mix, you put it on this mold, you allow them to dry for a few days, you take it out, and then there you have your three-dimensional structure. Now we created some pores in this three-dimensional structure. And how did we do that? We mixed some porogen. So in this case, we mixed some salt along with the polymer mix, poured that into the Teflon mold, and allowed it to dry. Once it was dry, we immersed those scaffold in water to allow the salt to kind of dissolve in water, and it dissolved away, leaving these pores behind. Now it's extremely important for these structures to have pores because it allows the cells to migrate, interact, and create that three-dimensional environment. So these are breast cancer cells. The second image there that you can see this round circular mass of cells which looks like a bunch of grapes. These are basically breast cancer cells forming a tumor. Now, why is it important to create these tumors outside of the human body? It is because we can use them as a drug testing platform, right? So instead of testing drugs on animals and then on humans straight away, we can test it on in these tumors first before we progress to human participants and etc. We can also use these tumors to study the progression of disease. Now, why this had not been done before? Because historically, we've been culturing these cells on flat, two-dimensional tissue culture plastic. And when cells are cultured on this flat, two-dimensional tissue culture plastic, they do not behave like they would normally behave inside the human body. Right? So when we provide these material-based three-dimensional scaffold system, the cells, which mimics the body structure or cell structure or cell system, the cells, they start to kind of behave how they would behave in a normal human body, and then express their phenotype in this way. So we use them to study different types of anti-cancer drugs. So this is how we've used these bacterial polymers to create scaffold and culture these kind of cancerous cells. The other application that we've explored is using them to create wound healing patch, for example. So there are lots of synthetic plastics used in making these, in developing these plasters. So we've used electrospinning systems, so to use these materials to kind of create. So we took this polymer solution, applied voltage, and we collected these fibers. This was again done in collaboration with a company in France called Fibrolane. They have this technique of impregnating active molecules in these fibers, and we use this to create this wound healing patch. Okay, so we've looked at 3D printed structures, fibrous structures. We've looked at tubular structures. We've also created these microspheres and nanospheres using these bacterial polymers. Now, what do these microspheres do? They act as a vehicle or a packaging material for drugs or active agents. They preserve the activity of the drug within them for a long period of time. So let's say, for example, age-related macular degeneration. It's a disease of the eye. So imagine a patient having to take injection in the eye every day, right? That could be very, very distressing. So what do these microspheres do? They take this drug, these drugs are encapsulated within these microspheres, injected in the eye. So instead of taking the injection every day, you would only take it maybe once in 3 months. So it continuously releases the drug over a long period of time while maintaining the activity of the drug. So we looked at anti-cancer drug and its release using this system. And this is the last application I'm going to talk about today. So this was a call that came out during the COVID period. What we developed was antibacterial, antiviral coatings for personal protective equipment. So we wanted to coat them, not just PPE, but also the hospital environment, equipment, catheter, etc., with antibacterial, antiviral coatings because they're very, very prone to bacterial and, of course, viral infection. So how did we do that? We took some, we chose some organic compounds, inorganic compounds, metal nanoparticles, etc., and we tested them against bacteria that are most commonly found in the hospital environment. For example, Staphylococcus aureus. I'm sure, even though you're architecture students, I'm sure you've heard of MRSA and the issues associated with MRSA and E. coli infection, etc. So we tested these active agents against these pathogens, and then we found that these compounds were 99.9% active against these pathogens. And then what did we do? We also created SARS-CoV-2 pseudotype virus. This was done in collaboration with my colleague, and we tested these active agents against these viruses. And if you look at this graph here, it's very simple to kind of understand. The percentage antiviral activity was within 80 to 90% for some of these compounds. What did we do next? We created these coatings. So we kind of mixed these active agents with our polymer using dip coating machine. We looked at their surface properties. They're very hydrophobic in nature. We then tested them against different pathogens. If you look at this plate here, you see these yellow blobs there. These are basically bacterial, these pathogens. And then the rest of the plate is basically empty because they were cultured on these our coatings, which means that it kind of killed almost 99% of the pathogens. And we found similar activity with the SARS-CoV-2 virus as well. So these coatings were active. So we are still working on different variants of COVID at the moment, and testing these coatings for different applications. So at the moment, we're thinking of PPE, but also of the hospital environment, etc. So just to summarize, we produce these polymers using bacteria. We characterize them in the lab. At the moment, we are working on scaling up these bacterial polymers with wonderful properties so that we can use them for large scale applications. We process these polymers into tubes, into fibers, into 3D printed structures, into spheres and nanospheres for various medical applications, and of course use them for, as medical implants, tissue engineering, drug delivery, coatings, disease modeling, and so on. So that is about my bioplastics, bacteria-derived polymers and their applications. But also just to tell you a little bit about our current project called the Cavendish Living Lab. This is now a vertically integrated project. We've got 3 live projects, basically. One is urban food growing system. So we are using the waste, converting that into biofertilizer and trying to grow food in unorthodox places like the university campus using hydroponic system. Early next year, our second live project is going to go live, which is bioplastics production. So if any of you are interested in making these bacteria-derived polymers and using them for different applications, please do get in touch. And we'll also be looking at water management, and there's going to be an element of artificial intelligence in all of these 3 live projects. We've already recruited people for the urban food growing system, and of course the most important bit is that this is a paid internship. So this is a project for 2 years, and William wanted me to share this with all of you. So I'd like to thank everybody who was involved in these projects with me, and all our funders, and of course our technical staff at the School of Life Sciences. So thank you for your attention, and I hope it was not too life sciencey for all of you. Thank you, everybody.
Thank you so much, Pooja. That was great, and there's a whole bunch of new things, including very good words like biocompatible, extremophiles, gyrospun fibers. I think we should be thinking about those in architecture. I wanted to ask you a question about the scalability of what you're doing, and obviously I understand that it's in a lab and it's not at a kind of building scale. But is there any reason it could be? I mean, is it scalable?
Yeah, it is definitely scalable. In fact, there are companies that actually have commercialized this process.
Yeah.
In comparison to synthetic plastics, we are not there yet. But of course, some of these systems have been commercialized. There are some companies. In fact, there's a UK-based company called Goodfellow. I think it's based in Cambridge somewhere. They do sell these polymers commercially. So if any of you would is interested to kind of process these polymers and see what they can do, you can. It's a bit expensive, but yeah, you can buy them. It's a company called Goodfellow. In the university, the bioreactor scale is 50 liters, for example. But then, if we collaborate with, let's say, Francis and Crick, they've got 100 liters or 200 liters, and there's this facility called CPI in north of England, I think somewhere in Middlesbrough. I don't remember exactly, but it took me a very long time to get there. When I went to see their bioreactors, and they had bioreactors of 1,000 liters, for example. So it is definitely a scalable process, and hopefully in future we can use them for larger applications.
Okay, thanks very much. And I mean, in terms of the manufacture of something, I mean, you've used 3D printing, but again, you could use all sorts of other things, couldn't you? I mean, if you were producing fibers, they could be woven fibers. Is that right? Or
Yeah, I think so. I think so. Well, so Well, for our kind of for medical applications that we've investigated so far, we've used electrospinning mostly, but we also have a melt electrospinning machine in our lab. So it's basically, instead of applying voltage, we melt the polymer and then kind of create those fibers. So that's another way that we can use. Injection molding as well. We've tried injection molding. So we've tried different technologies to process these polymers. I can see there's a question from Peter. How long do you expect the antiviral coatings to last? This is actually an excellent question. I'm just going to quickly go back and look at my slide just to make sure that, yes. So Peter, we've done an experiment with these coatings for 2 weeks, so 15 days. It does not seem like a very long time, but if you think about PPE, and from that perspective, 14 days is a very long time, right? So, so for 14 days, they seem to be kind of the active agent seems to be very active, but we need to do a long-term study to confirm if it would last longer than that. So we were thinking of kind of spray coating and things like that, but it's very early to kind of confirm that. But yes, at the moment, it seems to be active for 14 days. Thanks, Peter. Okay.
Yeah, thanks so much. Are there questions from the audience?
Any other questions? I'm happy to answer them. And if any of you would like to know more, have a tour of the facility, and have a look at the materials that we make, you can. I'll put my email in the chat here, so you can please write to me. I'll be happy to kind of host you in our school.
Thanks so much. I noticed Corinna said, yes, that'd be great. You should take your group down there, Corinna.
Oh yes, I'd be very, very happy actually.
Yeah, no, definitely. Yeah, I love it.
Sure, please do write to me, and then we can coordinate, make this happen.
Yeah. Great. Well, look, thanks so much for the talk. It's really, really, really fun. And you're a very good communicator. So I know you are talking science to a bunch of generalists, which are unfortunately architects. We're in this funny place between science and art or something. But I think it's so interesting, this field of new material, of inventing a material and defining and designing the properties of it. I think is really exciting, and especially as they're as they are biocompatible, which I think is a great term which I've never heard before. So yeah, that's brilliant. Thank you very much. Oh, look, there's a good question here. How much bacteria would you need to produce a bioplastic bottle of water? I think it's a wine there.
That's a very interesting question, Pietro. So in terms of bacteria itself, we actually start the process with a colony of bacteria. So just a kind of a pinch, just a single colony of bacteria, but then we provide lots of nutrients so they multiply and they grow. Like I said to you, so 6 grams per liter of culture, and we've got kind of 50-liter bioreactor, you can say. So if it's 6 grams per liter, for 50 liters, it would be kind of 30 grams. And that's quite a lot of polymer. I'm not sure how much polymer it takes to make a single bottle. I wouldn't be able to answer that. But if you're talking about bacteria itself, it's just a colony of bacteria, and we can kind of keep scaling that up to any quantity, any level that you'd like. Corinne has asked, given the state of our rivers in the UK, could you use hydrocarbon from the River Lee, for example? Actually, Corinne, that's a really good question as well. So I'm collaborating at the moment with my colleague who's a marine biologist. So we have isolated some really interesting strains from different places within the UK. We were trying, so there was actually a project called Antibiotics Undersea, which my colleagues had led, and you'd be very it was kind of shocking to find that, you know, we of course we isolate some really interesting strains, but at the same time we were analyzing water from rivers, Thames actually, and some of my colleagues reported that they found a lot of Coke contamination. So not the Coca-Cola Coke, but the drug Coke in River Thames, which is kind of interesting. But Karina, coming back to your question, yes, we are isolating lots of strains from hydrocarbon polluted areas as well. And again, the other interesting idea is to see whether these strains are able to degrade, considering the fact that they're able to thrive and grow in these hydrocarbon polluted area. We're also trying to see if they can actually degrade these plastics as well. So yeah, excellent question. I hope I answered your question. I'm not sure.
Oh, this is a good one.
If you have any views on the use of, this is something I've actually, Conrad, I've never thought about for green burials, but that's very interesting. I wonder if that's very interesting. Well, I've never thought about that actually, to replace gravestones. I think well, one thing to consider would be probably the degradation behavior and also the time span it would take for these green. Remember that there are different bioplastics that are commercially available. For example, polylactic acid, PLA, but they can only degrade under composting conditions. So they do not degrade naturally in the environment. PHA does. So, you know, in 6 months, they are completely degraded and gone. But yeah, that's very interesting. I actually hadn't thought about this at all.
Well, thanks again, Pooja. It was a really great talk. It was a really
Thank you.
lovely presentation. Yeah, thank you.
I hope it was not too sciencey, life sciencey.
Good. So just sciencey enough for us. Yeah. And yeah,
Okay.
we'd really love to have a tour. So a talk with Pete and Corinna, and maybe we can organise a bit of a group up there, if that's okay. That'd be.
Oh, of course, please do.
Yeah.
Thank you, everybody. Thank you for joining this session, and have a good rest of the evening, everyone.
Yeah. Thanks very much.
Bye, everybody. Bye,
Bye.
William. Thank you. Thank you, Natalie. Thank you.